Assalamu alaikum. This video will discuss Documents 5 of Chapter 9, Synapses, Structure and Function. By now, you have learned how a stimulus is converted by sensory receptors into action potentials, and how these action potentials propagate to the cell body and then down the axon in a unidirectional manner. But what happens when these action potentials reach the end of the axon, known as the axon terminals? What happens next? Actually, your nervous system is made up of a network of interconnected neurons. Every neuron makes connections with hundreds of other neurons. A neuron also makes connections with other body cells. The junction between the axon terminals of one neuron and another structure, whether another neuron or any other body cell, is called the synapse. And in this document, you are going to learn first about the different types of synapses and then about the function of these synapses. Document A of your books shows the different types of synapses. It shows neuronal synapses that are made between the axon terminals of a neuron and another neuron. Such synapses could be axodendritic if connected to the dendrites of a neuron, or axosomatic if connected to the cell body or the cell soma, or axoaxonal if connected to the axon. Other synapses are made between a neuron and a body cell, like a muscle cell, for example. But whatever the type of the synapse is, at the level of the synapse, you have to be familiar with these terms. One cell is known as presynaptic, where pre refers to before the synapse, and the other cell to which this neuron is connected is known as postsynaptic, where post refers to after the synapse, and the synaptic cleft refers to the area between the synaptic terminal of presynaptic neuron and the postsynaptic cell. At the level of the synapse, and whatever the type of the synapse is, scientists have noticed a common structure observed under the microscope, like those in Documents B of your book. Only the axon terminals of the presynaptic neuron contain bag-like structures called synaptic vesicles. Those vesicles are not found in the cell body or dendrites of the postsynaptic neurons. In order to find out the role of these synaptic vesicles and the function of the synapse, the following series of experiments described in paragraph 2 of your books are performed. In this experimental setup, two neurons are involved, a presynaptic neuron A and another postsynaptic neuron B. When neuron A is effectively stimulated, an action potential is observed at the axon of this neuron as well as neuron B. This indicates that the action potential generated at neuron A is transmitted to neuron B. But when the postsynaptic neuron B is effectively stimulated, an action potential is only recorded at neuron B but is not recorded at neuron A. This indicates that the action potential is transmitted in only one direction, from the presynaptic to the postsynaptic neuron and not the other way back. In the second experiment, when calcium ions are removed from the medium and neuron A is effectively stimulated again, an action potential is only recorded at neuron A, but not at neuron B. This suggests that calcium ions are indispensable for the synaptic transmission of an action potential from one neuron to another. The role of calcium ions is confirmed in the next experiment, because surprisingly, and in the absence of any stimulation of neuron A, and only upon the injection of calcium ions into the axon terminal of the presynaptic neuron A, an action potential is recorded in the postsynaptic neuron B, which indicates that the entrance of calcium ions into the presynaptic axon terminal induces the synaptic transmission of action potential. Now, in the third experiment, once again, without any stimulation of neuron A, the injection of a chemical called acetylcholine in the synaptic cleft between both neurons A and B causes the recording of a response in neuron B. This indicates that the signal is transmitted from the pre to the postsynaptic neuron via this chemical. But when the dose of acetylcholine injected to the synaptic cleft is increased, the screen of the oscilloscope connected to neuron B records the following. Notice that the depolarization in neuron B increases more each time. Of course, when this depolarization reaches the threshold, an action potential will be fired in neuron B. Now, based on the results of the previous series of experiments, scientists understood how a synapse functions, and they listed the sequence of events that occur at the level of the synapse upon the arrival of the action potential to the axon terminal. This synaptic transmission is illustrated in document E of your books and in the following video. At the level of the axon terminal, the arrival of action potential causes the opening of voltage-dependent calcium ion channels. The calcium ions flow to the inside of the axon terminals through these channels. These calcium ions will induce the fusion of vesicles that contain chemicals known as neurotransmitters with the cell membrane. This is how neurotransmitters are released into the synaptic cleft by exocytosis. The released neurotransmitters have specific receptors on the membrane of the postsynaptic neuron. The binding of these neurotransmitters to their receptors will modify the membrane potential of the postsynaptic neuron, or what we refer to as the postsynaptic potential. In the next document, we will see whether this postsynaptic potential will decrease or increase in response to the fixation of neurotransmitters to their specific receptors, and thus we will study the response of the postsynaptic neuron. But for now, let's focus on the neurotransmitters. At this point, it's very important to know that the fixation of neurotransmitters to their receptors is not permanent. Actually, they soon detach from their receptors. After that, some get degraded by specific enzymes found in the synaptic cleft, and others are recaptured by the presynaptic neuron by transporter proteins to be later used in another synaptic transmission. Assalamu alaikum.